Okay, everybody, welcome to our last chapter of the term. This is chapter 29, and we're going to be dealing with human development and genetics, and we'll essentially take it from fertilization all the way to old age, and then we'll talk a little bit about the impact that DNA has on the human condition with regard to physiology, physical traits, in some cases behavioral traits, and then we'll end the chapter with a discussion on the structure of DNA and where mutations come from. So we start with essentially the stages of life in prenatal development, and we note that from conception to birth, we generally break it down into three stages. The germinal period being defined as the first 2 weeks of development during the formation of the germ layers. The embryonic period is the second to the end of the 8th week, and this is important because organ systems develop during this period of time. That's followed by fetal development, which is the last 30 weeks. Well, what happens here is that the organ systems take on their fully differentiated state. Now, one of the things that we should discuss in this chapter is a couple of terms that I'll use in the description of the development of these organ systems, and they include morphogenetic movement, determination, and differentiation. And essentially, morphogenetic movement is simply the change in position of a cell or a group of cells from point A to point B, and this is thought to occur as a result of interaction with extracellular signals that are secreted by surrounding tissue. Differentiation is the change in a cell's appearance and behavior, and what usually happens during the course of development is that the cell's fate goes from a broad assortment of possible final adult tissue cells to one type of adult tissue cell. The reason that this happens is due to the way that the DNA is used in the developing cell. As it progresses through time, we restrict more and more of the genetic material so that the cell has its fate determined by the information that it can actually use. The other information is essentially chemically sealed off so that while it's present in the nucleus, it's not going to have an impact on the behavior of that cell. And then when we're talking about determination, we're simply discussing the interaction of a cell with chemical cues around it that act to restrict this genetic material and as a result direct that cell's fate. Okay, so we want to distinguish between those three things. Clinical age is defined as the mother's last menstrual period, and it's used to calculate the age of the unborn child. The post-ovulatory age describes the timing of the developmental events, and it's calculated as 14 days less than the clinical age. And you should remember why that is, because ovulation happens at mid-cycle when we're referring to, again, the interplay of the ovarian and the uterine cycles. Remember, it's that LH-FSH surge, which is facilitated in addition by the mechanical stroking of the fimbriae of the fallopian tubes across the surface of the ovary that results in the release of the egg. And so what we determine now for post-ovulatory age is the age of the child from the time of ovulation to the current time. Okay, the neonate is defined as from birth to 1 month postpartum. The term partum literally means birth. The infant from 1 month to 2 years. Child 1 to 2 years all the way up to puberty. Adolescent is defined as puberty, and this runs from age 11 to 14, anywhere within that time period, to an average of 20 years. Now, again, with a lot of these postnatal life stages, these are not hard and fast dividing lines. Puberty's onset is brought about primarily by changes in hormone levels that result in the emergence of secondary sex characteristics. And the onset can vary widely in individuals, as can, um, in many cases, the amount of time that these postnatal stages last. We see major physical and physiological changes and psychological changes that occur during this period of time. Onset in females, on average, between 11 and 13 years, in males, between 12 and 14. We should point out again, though, that there are exceptions to the rule. A condition known as precocious puberty can have onsets recorded as early as 5 years of age, and this is generally the result of abnormal secretion of hormones, gonadotropins, and of course the sex steroids that we've detailed in our discussion of the endocrine and reproductive system earlier than you'd see in the general population. It's followed by a period of rapid growth at puberty, which is then followed by a slower period of growth. Um, what spurs this rapid growth primarily is an increase in basal metabolic rate and in mitotic index brought about primarily by production of the sex steroids, which act as anabolic hormones in addition to the secretion of human growth hormone. Those are the two primary players that cause the increase in body mass and body height during this period of time. Remember that at the end of puberty, the epiphyseal plates in the long bones will close. Longitudinal growth from that point on is abrogated, and from that point on, the only change that you're going to find in skeletal components is either a change in density um, or a change in width. Usually, we reach adult stature by age 17 to 18 in females, by age 19 to 20 in males. On average, the males tend to be taller than the females, and this is due primarily to the influence of the increased levels of testosterone in males. The adult stage is defined from 20 years to death. Young adult dividing line around 20 to 40 years of age, middle age between 40 and 65, and the older adult, of course, the remainder of the lifespan from 65 onward to death. Okay, so we'll start at the beginning of a new human life, and what we have here is a diagram showing us essentially fertilization and the early events following. So we start up here in the upper left-hand corner. Many sperm cells reach the fallopian tube, and of those, a few dozen will attach to the corona radiata of a secondary oocyte. And during this trip through the female reproductive tract, remember that the sperms have become capacitated. So once they reach the corona, they're able now to release their acrosomal enzymes and digest through this outer protective layer. It's the combined action of all these enzymes that brings this about. Remember that a single sperm, in all likelihood, were it not accompanied by these other gametes, would probably not have a significant chance of fertilizing the egg. The head of a single sperm will penetrate the zona pellucida and attach to integrins in the oocyte membrane and penetrate the oocyte and enter its cytoplasm. So we can see this at this stage. The zona pellucida at that point will form a perivitelline space and prevents polyspermy by a couple of techniques. One is a cortical reaction that happens as a result of ionic changes near the surface of the egg that is intended again to lock out other gametes from entering. And then there's also a slower set of reactions that forms an impermeable barrier so that the sperm can no longer penetrate. But it's not to say again that those sperm that fail to penetrate have no role. They will help to move the egg down the fallopian tube towards the uterus along with the current that's created by the ciliated epithelial cells that line the uterine tube. So they have a role. In addition, of course, we have to acknowledge that their acrosomal enzymes ultimately made it possible for a single sperm to penetrate. Now, once we penetrate, the oocyte nucleus will move to one side. It'll then complete the second meiotic division and give off a second polar body. Remember that the polar body is a meiotic product that has very little cytoplasm and generally degenerates. When the second meiotic division is complete, we now will have the male and female pronucleus fuse, okay? And that's a process known as amphimixis. Um, this will result now in the production of the diploid state, and at that point, once the nuclei fuse, fertilization is defined as complete, and we now have a zygote, and we've now satisfied the criteria for a new and unique human life. The zygote will then rapidly divide. It'll form two cells about 18 hours post-fertilization. The two cells will divide to form 4 and 8, and so there'll be a geometric progression in the cell number. And we'll graduate now from a cell cluster to a ball of cells that we call a morula. Okay, it'll look a little bit like a raspberry. The cells in the morula, to a great extent, are defined as pluripotent. And what we mean by that is they're capable of forming a wide variety of adult tissue types. The reason for this is at this point in development, the genetic material hasn't been restricted so that the cell is unable to use it. And so the potential fates for these cells are relatively broad. The definition of a totipotent cell would be one that could form literally any adult cell type. And so what we'll see as we move through development is a progression from totipotent to pluripotent to further and further restriction of the cell fate until we get to what we call final differentiation. And at that point, as we move through this series of events where the genetic material is essentially sealed off from the cell as it moves through time and space, one of the things we have to appreciate is that once those doors have closed, the cell cannot go back and adopt another fate. And that's due, again, primarily to the covalent modification of the genetic material as well as other factors. After we generate the morula, we'll follow with the blastocyst stage. What will happen at this point is an event known as hatching, and will produce a hollow sphere of cells that will contain an inner cell mass that will eventually become the embryo and then the fetus. The blastocyst, upon its arrival in the uterus, will burrow into the endometrium in search of nutrition. The placenta will develop from the outer rim of cells, which we call the trophoblast, and it will eventually serve to nourish the developing fetus. Okay, so what you're looking at here are the early events, and you can see ovulation occurring mid-cycle. The egg is released. It is met by the sperm in the ampulla of the fallopian tube, and then by day 1, post-fertilization, we get to the 2-cell stage. A couple of days afterwards, more or less, we get to 4 cells. Morula by about day 5, and then by the time we get to the uterus, we have become now a blastocyst. The inner cell mass, which is going to be first the embryonic disc, then the embryo, and then the fetus, is shown attached to one side of this hollow ball. And then, of course, the trophoblastic cells are going to be the population that eventually protects the inner cell mass and also is going to act to essentially transport nutrients to what will eventually be the developing embryonic disc as it burrows into the endometrium. Okay, so this is the implantation event. What we're looking at here is a frontal section of the uterus and the uterine tube showing development 7 days post-fertilization. So we hang out in the uterus for about 3 days, and then we seek out the first well-vascularized tissue to attach to an implant. And the trophoblast begins to burrow into the endometrium forming what we call the syncytiotrophoblast. Remember that the definition of a syncytia is simply a population of cells that have fused together and are contained inside one single cell membrane. And what happens here is that proteolytic enzymes are breaking down the uterine wall, and what we're producing now are pools of maternal blood that begin to form. So here you can see at the intermediate stage of placental formation, which is between 2 and 3 weeks um, following, again, the fertilization event, maternal blood vessels are encountered by the syncytiotrophoblast, and they form these little blood-filled lakes called lacunae. This is the maternal blood that's going to contain the nutrients that will nourish the embryo. This is followed by the cytotrophoblast growing and surrounding the syncytiotrophoblast and also the lacunae, which are part again of the endometrial lining. And then we have embryonic blood vessels entering this range at about 1 month. And what you'll see once we get to full implantation and develop is that we've produced now by this stage a placenta. And what to notice about the placenta is that the fetal and the maternal blood do not physically mix. They're separated by the syncytiotrophoblast layer. However, they're capable of exchanging nutrients and waste products across this chorionic membrane, and that's what's going to nourish the developing embryo and fetus. In addition, the waste products will enter the maternal blood and be disposed of by the urinary system, as well as, of course, the digestive system. So what we have to appreciate here is that any molecule smaller than a certain molecular weight can easily cross this membrane and manage to get into the fetal blood. And this is one of the reasons why, especially during the first trimester, we warn mothers against drinking alcohol and taking recreational drugs, or being near individuals who might be harboring a pathogen of some kind, okay, especially a viral pathogen, which is small enough in many cases to cross this barrier and get into the fetal blood and will adversely affect development. So the mother's blood and the baby's blood in very close contact, but again, not mixing. The only point at which the maternal and the fetal blood will mix is actually at birth, when the placenta is torn from the endometrium, and that will represent for the first time an exposure between the mother and the baby's blood. And at that point, she will begin to develop antibodies against any antigens carried in the fetal blood. Okay, and we already talked about erythroblastosis fetalis as a consequence of this event. It happens when an Rh negative mother has an Rh positive child. That first Rh positive child, once it's born, its blood mixes with the maternal blood in the uterus that constitutes an exposure, and the mother begins to develop anti-Rh antibodies against the antigen on the baby's red blood cells. And then if there's a second pregnancy with an Rh positive child, there's the potential for those antibodies to cross again this chorionic membrane, invade the fetal blood, and destroy the red blood cells in an event known as hemolysis. We don't have this problem much anymore because we've developed a drug called RhoGAM, which essentially cloaks the Rh antigen, so that when an Rh positive baby is born to an Rh negative mother, if she's been on RhoGAM before, during, and after the pregnancy, the antigen will be masked from exposure to her immune system, and as a result, she won't generate antibody against the antigen, and she can have as many Rh positive children as she likes. But we need to be aware of the fact that, small molecules, small pathogens can cross this membrane and can adversely affect development. Okay, the germ layers mark a very important developmental milestone, because the germ layers are the cells that are eventually going to give rise, as we'll see, to all the adult tissue types. And what we see here is a look for the first time at the amniotic cavity. You can see it beginning to form here in blue just above. This would be the ectoderm, and this will be the endoderm. This will eventually be the yolk sac, and this will eventually be the amniotic sac that will surround and protect the developing embryo and fetus. At this point, however, um, the, the developing embryo is nothing more than a disc of tissue. We call this the embryonic disc, and it's made at this point of simply ectoderm and endoderm, which are part of the inner cell mass. The amniotic cavity, as we pointed out, is eventually going to form a protective fluid-filled bag around the entire embryo, and that's to provide protection against physical shock, but it also helps to maintain homeostasis. The yolk sac you can see down here forms inside the blastocele primarily from the endodermal cells, and the yolk sac will serve as a temporary source of nutrition for the developing embryo while we establish the structures that will eventually make up first the chorion and then ultimately the placenta and the umbilical cord. Okay, so what you're looking at here is just the sequence of events that takes place after the embryonic disc is formed. And for the first time, we see morphogenetic movement playing a major role here. There's been morphogenetic movement up to this point, but this is a landmark event. What you see is that cells in the epiblast are going to move toward this little groove that's created along the dorsal side of what's eventually going to be the embryo. And they're going to migrate through this primitive streak and populate a layer between the endoderm and the ectoderm. These cells are defined as mesodermal cells. Okay, so they're indicated here in the diagram in red, the endoderm here in yellow, and the ectoderm in blue. You can see the developing yolk sac below and the developing amniotic sac above the embryonic disc. Now, what's the significance of this event? Well, one of the things that we have to appreciate is that this establishes for the first time a, an anterior-posterior axis. The head of the embryo will eventually be somewhere around here, and the end of the spinal cord will eventually be somewhere around here. It also provides an axis of bilateral symmetry so that we have structures that are mirrored on the left and right side of the developing embryonic disc for the first time. The notochord, which is going to develop shortly after this, is essentially a column of tissue that will develop underneath this primitive streak and will serve to induce the formation of a neural tube. And the neural tube is eventually going to be the spinal cord, the brainstem, and the brain. You can see the three germ layers here at midline in the disc. We're looking here essentially at a sagittal section, so to speak, really a parasagittal section. So we're looking edge on at the developing disc. And you can see the embryo proper defined by these three germ layers. Now, next thing that's going to occur is that a neural plate will form along the ectoderm, okay? And this is induced by the presence of the notochord just underneath the neural plate. It's thought that chemical signals from the notochord produce these changes in shape and movement in the cells directly above it. The neural folds will form parallel ridges along the embryo. At this point, we have a neural groove and a crest up here. We call this the neural crest, the neural groove, and this whole thing is the neural fold. And notice again the notochord persisting just underneath this structure. The neural crest cells then begin to form, um, and they'll eventually jump off the neural crest and then migrate um, into the middle layer of the embryo, where they'll produce a wide variety of structures, among them craniofacial structures, portions of the developing heart, and other components of the nervous system, as we'll find out shortly. The neural folds will eventually meet at midline and form the neural tube, and the neural crest cells will separate from the neural fold and begin to populate now what will eventually be these other adult structures. So what we're seeing here is two things going on at the same time. We're seeing morphogenetic movement because we're seeing a change in the shape of this structure of the embryo, and we're also seeing a change in the fate of these different cells. Okay, we know, for instance, that the neural tube is going to eventually be most of the central nervous system. We know that the neural crest cells are destined for a wide variety of fates, and you can also see that the overlying ectoderm will eventually produce the skin. Now, one of the very interesting things also about this point in development is to notice that along the neural groove, okay, you can see the tube developing from caudal to rostral. So the first thing really to begin to form is the portion of the neural tube that will develop into the spinal cord. The next portion, the portion that will develop into the brainstem, and the final portion of the neural tube will form the brain proper. The last feature that will show up as a central nervous system structure will be the telencephalon, which will eventually give rise to, among other things, the cerebral cortex. Now, why is this such an interesting sequence of events? Well, it's due to the fact that um, we can see in human development, in the process that brings about the adult organ systems, events that seem to mirror evolutionary events. And so there's a term for this that biologists like to use: ontogeny recapitulates phylogeny. And in the production of the spinal cord, the brainstem, and the brain, we see a progression of the nervous system structures from primitive to more advanced in terms of an evolutionary relationship. So if we look at, for instance, at very primitive animals such as an earthworm, we see a sort of central nervous system, but we don't really see much of what we would appreciate as a brain. Essentially, we just have ganglia at the cephalic end of the animal. And then as we continue to move up the evolutionary ladder, we see the emergence of a brain stem. If you've ever looked, for instance, at the brain of a frog or of a fish, it looks very much like a human brain stem. And then we don't really see a cerebral cortex, an extensive cerebral cortex, until we get to the mammals. And at that point, we see an elaboration of that structure as selection has brought about a greater need in those organisms for cognitive development. Okay, so we go from primitive to advanced in evolution. We go from primitive to advanced here in the production of a new human being. And what that seems to indicate to biologists is that the memory of evolution is somehow contained in our genetic material. The neural crest forms when the sides of the neural tube meet at midline. Some of the cells will break away and migrate along the side of the neural tube, and what will they produce? Well, a lot of the postganglionic portions of the peripheral nervous system, a lot of the sensory apparatus, the adrenal medulla will form from these cells, the melanocytes in the skin, and portions of the craniofacial structure, parts of the skull, the dentin of the teeth. Some skeletal muscles will form from neural crest, and we'll also see mesenchyme in the cephalic region coming from these cells as well. Now, this is just a chart showing you the germ layer derivatives in great detail. Um, Basically, if you want a thumbnail sketch of what forms what, the ectoderm is responsible for the bulk of the nervous system and the skin. Um, the mesoderm is responsible for the musculoskeletal and cardiopulmonary system. And the endoderm forms primarily um, the internal organs, the lining of the gut, and so on. That's a bit of an oversimplification, but that sort of gives you an idea of what structures come from what. Okay, so that's table 29.1 in your book. Now, one of the things that we'll notice the minute that we establish an anterior-posterior axis and bilateral symmetry is the emergence of somites. The neural tube will begin to continue development, and mesoderm next to it, lateral to it, will form little masses of tissue that we call somites. And you can essentially track the development of the embryo at this point by counting the number of somites in the embryo. The fewer somites, the earlier in development we are. These will eventually become portions of the skull, the vertebral column, and the skeletal muscle. The gut and the body cavity are the purview of some mesodermal components, but mostly endoderm. The embryo is going to become essentially like a tube within a tube from this point out, and we'll begin to see the emergence of a foregut and a hindgut. Um, the foregut developing at the cephalic end, which is where the head's going to be, and the hindgut near the caudal end, which is where the end of the spinal cord is going to be found. The developing digestive tract will pinch off from the yolk sac as a single tube, but it'll remain physically attached at its caudal and cephalic ends for most of the rest of its development. The ends of the fore and the hindgut also maintain contact with the overlying epithelia, and they'll form an oropharyngeal membrane up near where the mouth is going to be located, and a cloacal membrane, which is going to become eventually the anus and the urethral opening. And again, here we see an interesting primitive structure forming. The cloaca is essentially a primitive opening for the excretion of waste. Where do we find cloacas? We find them in reptiles and in birds, and we don't see a significant separation of these two duct systems until we start to get up into the higher mammals. Okay, so let's talk a little bit about the structures that are associated with the gut. So we're looking here now at the internal organs that compromise the digestive system. Evaginations are defined as outpocketings from what will be a tube that'll run from the mouth to the anus, as opposed to an invagination, which is what we saw with the formation of the primitive streak and eventually what would become the neural tube. The allantois will become part of the bladder. Other evaginations will form the pituitary and thyroid, as well as the lungs, liver, and the pancreas. Branchial arches are bars of tissue along the lateral surface of the head, and the sides of the gut will expand to meet them to form pharyngeal pouches. The central pouch will form the pharynx, and the others are going to become the auditory tubes, the tonsils, the thymus, and the parathyroids. Now, what's significant about this is that the branchial arches resemble, to a great extent, gills. And so this is another example of a primitive structure that will appear and disappear as development proceeds. In addition, at this time, body cavities are going to begin to make their presence felt. We define a body cavity, a true body cavity, as a coelom. It's going to first fuse around the heart, forming the pericardial cavity, and then it's going to expand backwards to become the pleural and peritoneal cavities, which will eventually separate from each other at the border formed by the diaphragm. You can see here in this essentially developmental sequence, you're looking here at a sagittal view of the developing embryo, and this would be a transverse view. Okay, so a sagittal section through here, transverse section through here. And again, what you're seeing is two things, right? You're seeing morphogenetic movement, but you're also seeing differentiation at the same time. So what we're looking at here in the early stage of the formation of the digestive tract is the yolk sac is going to begin to shrink as the embryo becomes more and more dependent now on nutrition from the maternal blood, and it will be essentially it'll eventually atrophy, and the only remnants are going to be components that will form parts of the digestive system of the tract, and also small components of the urinary system forming portions of the bladder. Okay, but you can see here that as the ectoderm is expanding both caudally and rostrally, we're beginning to enclose what will be the yolk sac, and it's beginning to shrink more and more. What will happen, as you can see here, is that mesoderm is going to begin to impinge here. You can see it growing towards towards midline, both caudally and rostrally, and it's eventually going to pinch off and form now the true coelom. It basically will close off a portion of the yolk sac. You can see here in transverse section, it'll wrap around, and this is going to be the lining of the gut right here, okay? And the mesoderm will form, among other things, musculoskeletal components, and here again you can see, of course, the neural tube, which is eventually going to be the spinal cord, and eventually the mesoderm is going to ensheath that in the vertebral column and in the bones of the cranium. Okay, so you can see in transverse section how the coelom is beginning to expand laterally to the tube that is going to make up essentially the GI. Limb bud development begins at about 28 days. You can see small protrusions that will eventually be the arms and legs. There will be an apical ectodermal ridge that will develop on the lateral margin of each limb and stimulate outgrowth. The limb tissue is laid down from proximal to distal. So the last thing to show up are the fingers and the toes. The first to appear are going to be the structures that will make up the upper arm and the thigh. Okay, and we've detailed extensively components that can interfere with limb development. There are certain viral infections that are associated with abnormalities in limb development that result in increased or decreased numbers of digits. There's a very famous instance of thalidomide, which was marketed initially as a morning sickness drug, producing a condition called flipper limb, in which the long bones of the arms and the legs fail to fully develop, and sometimes you'll end up with the carpals fused directly to the humerus in many cases. It was thought initially that this was a genetic condition when it was first recognized in the 60s, but as a result of statistical studies, they were able to link this condition, flipper limb, to the use of thalidomide as a morning sickness drug, and it was taken off the shelves for many years. It's now reemerged as a treatment for conditions such as cancer and leprosy, but it's only given to postmenopausal women and males. So this drug is now back in vogue. Here you can see the events that lead to facial development. 28 days post-fertilization, the face is going to develop from three processes that radiate towards the anterior aspect of the embryo. You're going to have a frontonasal process that's going to eventually form the frontal bone and portions of the bridge of the nose and the nasal cavity. You've got maxillary processes that will eventually fuse to form the upper jaw, and mandibular processes which will of course form the lower jaw. 33 days post-fertilization, the nasal placodes appear on the frontal nasal process. So you can see those here and here. Okay, those are going to eventually form the nostrils, and they'll move towards midline at 40 days post-fertilization. The maxillary processes extend towards midline. The placodes move towards midline and fuse with the maxillary processes, and they're going to form the jaw and the lip. Okay, so you can see now how this proceeds. 48 days post-fertilization, continued growth brings everything back towards midline, and you can see at 14 weeks post-fertilization, essentially the configuration that we're going to have in the adult. Now, this is a very interesting process because what you're seeing here is a developmental sequence of arch closure. Okay, we've seen it once already in the formation of the neural tube, and we're going to see it also in the formation of the vertebral column and of the bones of the cranium. And now we're seeing it in the facial bones, forming a lot of the facial structures. Arch closure failures can produce a wide variety of birth defects, among them spina bifida, where the spinal cord and the associated membranes protrude dorsally out the back of the body. Also another condition known as anencephaly, where the brain fails to fully form inside the cranium. This is often associated with stillbirth. And then another failure of arch closure you can see here with facial development, which is the condition known as cleft palate. Cleft palate can present as minor a condition as hair lip, where we have a minor defect in the midline anterior aspect of the maxilla. It can also present as severe as a single opening in the middle of the face for both the nose and the mouth. It's thought to be correlated in some cases with a lack of B vitamins in the diet of the mother, because we see a higher incidence of these conditions in mothers that either have a problem absorbing the B vitamins or that just lack them essentially in their dietary intake. Okay, so that's the significance of that developmental phenomena. Okay, now some more organ systems for us. The skin is derived from the ectoderm. The dermis comes from the mesoderm, or of course neural crest cells, which form a lot of the craniofacial structures. So here again, we see two different embryonic tissue types producing now a single organ, right? The skin. The skeleton develops from mesoderm, and in some cases there will be neural crest contributions. Muscle comes from myoblasts that are early embryonic cells that will develop into skeletal muscle. And the nervous system, of course, is derived from neural tube and neural crest cells. The neural tube, most of the central nervous system structures, the neural crest, many of the peripheral nervous system structures. Okay. Let's touch for a little bit on the development of the special senses. We should note that the olfactory bulb, the first cranial nerve, and its associated tracts are evaginations of the telencephalon, which will eventually form the cerebral cortices in the central nervous system. The eyes are evaginations from the diencephalon. They eventually become the optic stalk in the vesicle, and the vesicle will induce lens formation once it reaches the epidermis. Again, this is an example of a chemical signal that's being secreted by these structures in order to now produce a change in appearance and behavior in a nearby tissue. In the ear, we'll see an ectodermal placode invaginate and then pinch off. What we should note, again, about the origin of these structures is the correlation of the appropriate cranial nerves to their relevant structure. Remember that the olfactory bulb is found just underneath the frontal lobe. It sits atop the nasal cavity above the ethmoid bone, and it essentially brings action potentials relating to our sense of smell, ultimately back to the frontal lobe, okay, uh, where we will interpret that sensation as a particular type of odor. We know that the optic nerve is closely associated with diencephalic structures. Remember, the diencephalon consists of the epithalamus, the thalamus, and the hypothalamus, as well as the um, associated glands, the pineal gland and the posterior pituitary. Um, and so it's not a surprise that the optic nerve is associated with these structures in the diencephalon, since we now know that it originates from the diencephalon during human development. Okay, the cardiovascular system begins really with the formation of blood in areas that are near the yolk sac. The vascular system and the heart don't form until later in development. A heart actually begins as two beating tubes that fuse at anterior midline, and as we'll see, they undergo morphogenetic movement to generate now a four-chambered pump. The respiratory system begins as buds that evaginate from the foregut near the esophagus, and the urinary system originates as um, kidneys that originate from mesoderm between the somites and the lateral portion of the embryo. The reproductive system, which is closely associated with the urinary system, is going to produce the male and female gonads, which begin as gonadal ridges and are closely associated with primordial germ cells that will eventually become the male and female gametes, the oocyte and the sperm. And we'll deal with those developmental processes in the next podcast. Let's take a look at heart development, as it presents now another example of how ontogeny recapitulates phylogeny. Note up here that at 20 days post-fertilization, the heart consists of two beating tubes that fuse at anterior midline into a single beating structure. 2 days later, the fusion is complete, and we form a single beating tube which will begin to bend as it elongates. And so you can see sort of a twisting motion here indicated in the middle portion of the diagram. This happens, of course, in the pericardial space. 31 days post-fertilization, the interatrial septum and the interventricular septum begin to grow towards the center of the heart. So what we have here is a transition now from a one-chambered pump to what will be a two and then a four-chambered pump. As we continue, 35 days post-fertilization, the interventricular septum is almost complete. A foramen is going to open up in the septum premium as the septum secundum begins to form. So you can see the septum premium here indicated by this blue disc, and the septum secundum essentially migrating again towards the midline of the heart. Final embryonic condition of the heart will have the interatrial septum featuring the foramen ovale, which allows blood to be shunted from the right to the left atrium while the fetus is still in utero. After birth, the blood will begin to flow in the opposite direction. The septum premium will be forced against the septum secundum, and this will now close the foramen ovale, and this will now restore the adult circulatory pattern. So you can see how initially these two regions of tissue are separated and allow now continuous flow of blood from the right to the left atrium, but eventually they'll close off. The purpose of this is to allow oxygenated blood to get to the pulmonary structures as well as to the rest of the body of the embryo. And we'll elaborate on this when we talk about fetal circulation in the next podcast. Here we're looking at lung development, 28 days post-fertilization. a bud is going to form that splits into two buds that will eventually become the lungs and primary bronchi. You can see that here. This is the foregut, and this is the evagination that's going to become now the future trachea, and these will be eventually the primary bronchi and the lungs. At 32 days post-fertilization, the primary bronchi branch into secondary bronchi. These will supply the lung lobes. And then at 35 days post-fertilization, the secondary bronchi branch into tertiary bronchi. These will supply the bronchopulmonary segments. And at 50 days post-fertilization, the branching will eventually form the respiratory passages in the lungs. Okay, so you can see here how the tracheobronchial tree has become more and more elaborate as development proceeds. Okay, we will pick up with the development of the reproductive system next time we're together, and I'll see everybody in class. Thank you.